Hey guys, so here we are for concept 10.2. We're going to get into the details of the two basic parts of photosynthesis. If there's anything good, as far as good news here, at least we can say that photosynthesis really only has two major portions to it, as opposed to respiration, which had three major portions. You're going to find a lot of similarities, or a lot of things that tend to look alike or act alike between respiration and photosynthesis, even though these two overall processes are kind of doing the opposite of each other. One of them is breaking down molecules and releasing energy, and one of them is taking in energy to build organic molecules. They still use a lot of the same sort of pathways, just kind of in a reverse way. So concept 10.2 says the light reactions, which is the first part, convert solar energy to the chemical energy of ATP and NADPH. So we're going to use sunlight and water, and we're going to use those materials, and when we're done with this first portion, the light reactions, we will have ATP and we will have some NADPH. And those energy-laden molecules will then be used in the second portion called the Calvin cycle. Now we're going to see NADPH. That may look somewhat familiar to what we've seen before. Again, both of these two processes in Chapter 9 and 10 are kind of interrelated to each other, of course, at least in the plants. NADPH, which is formed from NADP+, is very similar to NADH, which was formed from NAD+ in respiration. So NADPH is basically in photosynthesis what NADH was in the respiration. It's just an electron carrier. It's what I would have been calling a temporary battery, or a bucket, basically, to carry these excited electrons from one part of the process on to the next. So back to our details here. So we're looking in the chloroplasts, which are a part of the cell, a cell organelle only found in the plants or the other photosynthetic producers. Chloroplasts are basically solar-powered little chemical factories, and the product they're churning out are the ATP and NADPH molecules. So their thylakoids transform light energy plus water and some other basic products into ATP and NADPH. We know that ATP is the energy currency of all cells, and NADPH is, again, just another one of those electron carriers that's used as a temporary battery or a temporary storage or a carrier molecule for these excited electrons as part of the process. Let's talk a little bit about sunlight and some physics, just a little bit, to kind of understand where we're going here. Light is a form of EM energy, electromagnetic energy, also known as electromagnetic radiation. There's a huge variety of electromagnetic energy or waves. Light is a very interesting material, or I guess form of energy, we should say, because it actually has a dual nature. It actually can sometimes act like a wave and sometimes act like a particle. In this case, we're going to be looking at the wave tendency of light. And light has, like all EM radiation, has a certain wavelength, which is the distance between the crests of these waves. And the wavelength determines what type of electromagnetic energy we're talking about here. And EM waves can have a huge difference in size or wavelength. The smallest wavelengths, the smaller the wavelength, the more powerful or energetic the wave, and the smallest or the shortest distance of these EM waves can be less than a nanometer. That would be like gamma rays, very harmful radiation to living creatures. And the longest of these waves are actually radio waves, which can be more than a kilometer long. So you can have waves that are almost or more than a kilometer long, approaching, close to a mile in length. And then you can have some waves that are less than a nanometer in length. When we talk about the entire electromagnetic spectrum, we're talking about everything from the very long radio waves all the way to the very short, highly energetic, what we would kind of consider radioactivity, the gamma rays and things like that. What we can see is just a small portion of that. We call that small portion visible light, and that consists of the wavelengths that our eyes can actually detect and see, that produce colors that we can see with our eyes. It's also the wavelengths that drive the photosynthetic process. So we mentioned that sometimes light behaves as a wave, sometimes it behaves as a particle. When we think about it as a particle, we call these particles a photon, and each photon of light carries a specific fixed quantity of energy. And so the photons of light carry differing amounts of energy depending on their wavelength. And actually, this dual nature of light is very interesting if you're a physics type of person. It's been studied by many scientists, including Albert Einstein, who won his Nobel Prize for his study of this duality of light and what's called the photoelectric effect. Something perhaps to look up on your free time or ask about in physics class. So here we have the entire EM spectrum, going from the largest waves, the lower energy waves down here on the right, to the smallest high energy waves on the left. And within there is a small little segment that we call visible light. And within that visible light, there's a spectrum of colors that our eyes see. And those colors differ based on the wavelength of the light. And it also gives you a different amount of energy per wavelength. So the longer the wavelength, the lower the energy. The shorter the wavelength, the higher the energy. There's actually a little saying, Roy G. Biv, ROYGBIV. So that would be red, orange, yellow, green. Man, I got to get it straight. Red, orange, yellow, green, blue, indigo, violet. Roy G. Biv, if you want to try to memorize the colors from the longer wavelength, lower energy, all the way to the shorter wavelength, higher energy. Within the plant, especially within the chloroplast, there are these pigments. And a pigment is basically just a substance that absorbs one wavelength or multiple wavelengths of visible light. And different pigments absorb different wavelengths. And the wavelengths that are not absorbed are reflected. In other words, they bounce back, or they're transmitted, meaning they pass through. The reason that leaves and plants appear green, for the most part, is because chlorophyll will capture wavelengths of light except for the green wavelengths. And those green wavelengths are the ones that either bounce back or pass through. And so that's what our eyes see, are the waves that did not get absorbed. So the color of an object is the wavelength of the light that's either reflected or transmitted, and that's the color that was failed to be absorbed, the wavelength that did not get absorbed. Here's a quick animation to kind of give you some details about light and this absorption or reflection of different wavelengths. You can hit right-click and hit play, and you should be able to see the animation. Then here's a still photo if you didn't get to see the animation or you just wanted another way of looking at it. Again, the reason that leaves look green is because the chlorophyll inside of them, the chlorophyll will absorb almost all of the various wavelengths except for the green wavelengths. And that light either gets reflected or transmitted all the way through. And so that's what our eyes see. So when you see, say, someone wearing a red t-shirt, that the pigments that were used to color that t-shirt absorb all of the wavelengths except for red. And the red is what is being bounced back. And that's why in our eye or in our brain we see red. So then if you really want to get into some head-scratching information and kind of things that make you think, is to think about white light, which is a combination of all the wavelengths, and then what is black. And if something is black, what does that mean, and how can we see black? But again, that's into physics, and that kind of gets us off topic. So that's what we need to know, and we'll move on. We'll take a look at some quick history about how this information was discovered. There's a device that we can use called a spectrophotometer. And a spectrophotometer is basically measures the ability of a material to absorb certain wavelengths. So the way that that's set up is you'll have a light source that produces many wavelengths. So it may produce all of the visible wavelengths of light. That light passes through a prism, which breaks it up into its constituent parts. So then you have a particular slit or an apparatus here that will slowly move and will only allow one little set of wavelengths at a time to pass through. So first you may get the red, then the orange, then the yellow, then the green, all the way down. And those individual, basically beams of wavelengths, particular color, will pass through your sample, and then it will hit a detector. And the detector will measure activity and tell you how much of that particular wavelength was absorbed or either reflected or transmitted through. Here's another kind of picture showing the setup. So what happens is you have this detector that measures the amount of light that's coming through. And so, for example, when this is allowing the green light through and there's chlorophyll in the solution, well, the chlorophyll does not absorb very much of that green at all. So the vast majority is reflected or passes through. and that causes a spike in what's called the galvanometer. Well, that's a hard one. Galvanometer. So it's allowing a large amount of light to get through, a large amount of energy, because it's very little absorption, and so it pegs out the galvanometer. Down here, when we move the slit to allow the blue light through, well, a larger amount of the blue light is absorbed by the chlorophyll, so very little gets through, and you can see that shows very low on the dial here as far as amount of light getting through. So using this, we can see, okay, what does chlorophyll absorb and what does it allow to either be reflected or transmitted on through. Using that information, we can make a chart basically called an absorption spectrum. It's kind of like a chart or a graph that shows us the different wavelengths of light and how much is absorbed based on any particular wavelength. Chlorophyll kind of comes in two flavors, you could call it, chlorophyll A and chlorophyll B. Chlorophyll A works best or absorbs most in violet, blue, and red light. So that's where you're going to get the most bang for your buck, basically, as far as the wavelength of light. You can also create a chart called an action spectrum, and an action spectrum basically just shows the effectiveness, or again, kind of like how much bang for the buck are we getting for a particular wavelength, in this case for photosynthesis. So if you want to look here, we have a couple of different things. We have the absorption spectra, and we have a couple of different materials within the cell, chlorophyll A, there's another type of chlorophyll called chlorophyll B, and then there's some other colors that we don't see as often, but they're listed here as well. They're called the carotenoids, and we do see these oftentimes in the fall, oranges, yellows, different colors of pigments, and you can see the wavelength of light that they best absorb. And then down here we have an action spectrum, which is where are we going to get the most photosynthesis at what point. So you can see they kind of line up with the absorption spectra. You're going to get high points of photosynthesis using this wavelength and this wavelength of light. And this last little chart talks about an old experiment that we'll discuss just briefly. So before we had this fancy equipment to kind of monitor and look at this, how did we first learn about this? Well, it's kind of an interesting experiment that was used. So let's take a look at that. The action spectrum of photosynthesis first was kind of discovered or demonstrated way back in the 1800s by this scientist named Theodore Engelmann. And he didn't have the type of equipment that we have now. So the way that he did this was using a type of algae. And he had these long strands of algae, and he would expose different parts of the strands of algae to different wavelengths or colors of light. And he also had mixed in with the algae samples some bacteria. And so his thinking was where the plant used the wavelength the most and did photosynthesis the most, it was already known that photosynthesis released oxygen. So there'd be the greatest oxygen output at certain wavelengths of light. And he thought, okay, the bacteria need the oxygen. They would accumulate in those areas as well. So he was growing aerobic bacteria, which need that oxygen, and they would tend to accumulate along the strand of algae where the oxygen output was the highest. And that oxygen output would be the highest, of course, in the areas where the wavelength of light allowed for the greatest amount of photosynthesis. So looking back at this chart, he was measuring how much bacteria kind of gathered or reproduced in certain areas. So he had a long strand of algae, and he was shining different colors, bands of light, wavelength in different areas, and he had spikes in the amount and number of algae in this, bacteria in this location, in this location. And sure enough, that pretty much lines up with our modern day equipment readings that we see here. So he was using bacteria kind of as a biological mechanism for discovering this or showing this. Kind of like a living spectrophotometer, basically, is what he was doing. We mentioned that there was a couple of other chemicals in there. Chlorophyll A is kind of a blue-green colored chemical. That's the main one that we're going to be looking at. There's also some what we call accessory pigments. One of them is called chlorophyll B, and it's more of an olive green color to our eyes. It helps allow the plant to capture more light energy and use it more effectively. And there's also some other accessory pigments that we don't often see, except usually in the fall. These are called carotenoids, and they're going to range from yellow, orange, red types of colors. They actually have some activity within the plant even during the growing season when the plant looks just green. Those carotenoids are in there. They help do things like absorb excessive light that can damage the chlorophyll and damage the chloroplast, and they have a few other purposes as well. We don't often see those colors, but we do when the plant starts to shut down for the fall. It stops photosynthesis, stops producing chlorophyll, and then suddenly those carotenoids show up and we see them in all the beautiful fall colors. Chlorophyll A and chlorophyll B, very, very similar as far as their chemical makeup. Really, the only difference they have is as part of this one ring structure. In chlorophyll A, there's a CH3 unit here, and in chlorophyll B, there's a CHO. We don't have to memorize that. I just wanted to show you to tell you that that one change makes a difference in the color of the chlorophyll that we see with our eyes, and it makes a difference in the wavelength of light that it absorbs. So it's kind of odd that one tiny little change in a very complex molecule has such an effect. Don't have to memorize that. You would need to know chlorophyll A is the primary one, and then chlorophyll B and the carotenoids are some of these, what we call accessory pigments. So in these various pigments, when sunlight hits them, the photon of light basically imparts energy to these pigments, and it basically excites their electrons. And when they absorb this light, their electrons go from a ground state, which is a more stable, low energy state, to a higher excited state, which is kind of unstable. And when these excited electrons kind of fall back to where they belong at the ground state, they re-release that energy, oftentimes as light itself, photons. And we call this phenomena fluorescence. So if something fluoresces, it's absorbing energy, exciting electrons, then those electrons fall back to where they belong, and they give that energy back off as light. So if we had a solution of chlorophyll all by itself, we separated it out from the plant, and we just had the chemical chlorophyll like in a petri dish or in a jar, if you put light on it, it will fluoresce. It gives off light and heat. It absorbs that energy. It excites electrons, they fall back to where they belong, and that energy is given off again as light and heat. And it's kind of weird because it actually gives off light or fluoresces as a red color. So here it is. Now this is not within the plant, but chlorophyll, pure chlorophyll, apart from the rest of the chloroplast and everything else, will fluoresce in this kind of interesting blood red color and gives off heat as well. To excite these electrons, to get them to jump up to this particular excited state, requires a set amount of energy to make them jump to a set place. And that amount of energy is different for different molecules, and that's why the different pigments absorb different wavelengths of light. They'll only absorb the wavelength that provides the correct amount of energy to make their electrons jump the particular amount up to their excited state. So different pigments absorb different amounts of energy, which is different wavelengths of light. Chlorophyll fluoresces with this bright red. Doesn't do that in the plant though, because that energy is not going to be re-released as light and heat. It's going to go toward other processes. So within the plant cell, within the chloroplast, there are systems called photosystems. And a photosystem is basically a pretty complex system of proteins and molecules that all work together to harvest light and create these energy molecules, ATP and others, from this light energy. So the photosystem consists of basically two parts. You've got the inner portion, which is called the reaction center, and it's a type of protein. It's in the middle. It's kind of like the bubble gum inside the Tootsie Roll or the peanut inside the peanut M&M. And then that's surrounded by a light harvesting complex. A light harvesting complex is the pigment molecules that we've been talking about bound up with some other proteins. They capture the energy from the light and transfer that energy into the middle to the reaction center complex. So here's a great picture. So here we have a lipid bilayer. This is the thylakoid membrane. So we're in the chloroplast looking at the thylakoid. This is the thylakoid membrane. Light is coming in, and surrounding on the outside here we have, or this entire thing is called the photosystem, but surrounding the outside is the light harvesting complex. The light comes in, it excites electrons. Those electrons, the excited electrons, basically just get passed from one pigment molecule to another. The energy is transferred until finally it's transferred into the middle portion here, which is the reaction center complex. In this middle portion we have some very special chlorophyll A molecules, and then we have what's called a final or primary electron acceptor. So the energy is first captured on the outside here by the various pigment molecules, and that energy is then basically transferred back and forth to all these different pigment molecules in a long chain until it's finally given into the middle to the reaction center. And then that's where some of the important activity we're going to look at occurs. If we wanted to see more of an actual sort of model of what it might look like, remember these are proteins, so they're very complex. There's all sorts of shapes and folds and bends and turns, and that shape and all the folding is very important. So within, we have chlorophyll molecules connected to all these various proteins that make up this photosystem that we're going to take a look at. In the middle of that photosystem, we talked about there's something called the primary electron acceptor. Right there. That's where those energized electrons are eventually heading, to that primary electron acceptor. That's what accepts the excited electrons, and it gets reduced, which means it goes lower in negativity. And then that transfer of electrons, that transfer of energy from the chlorophyll A molecule to that primary electron acceptor, that's the first step of the light reactions. So as opposed to a test tube of chlorophyll where it will give off that energy as heat and light, that's not going to occur. That energy gets captured by this electron acceptor, this primary electron acceptor, and it's going to be used later on down the line. Now, there are two photosystems that work together to achieve this whole process. So in step one of photosynthesis, where we're capturing light and we're forming ATP and NADPH, we're going to need to use two photosystems, two basically light gathering systems. Now it gets kind of confusing because the first one in the chain is called photosystem 2 or PS2. And I hate that it's still called PS2 or photosystem 2 because it's actually the first in the process. But it's called photosystem 2 because it was discovered second. It was not the first photosystem discovered and found. Why they haven't changed it and just changed it to be like a linear order, I think sometimes it's just to make biology students cry. But the key thing to keep in mind is that photosystem 2 is actually the first photosystem in our long chain of events. Photosystem 2 is also called PS2 or P680 because 680 nanometers is the wavelength that it best absorbs. Now, 680 nanometers would be in the red. As far as our eyes, that would be kind of red coloration. The second photosystem in our chain here of events is called PS1, or photosystem 1. Again, I'm very sorry that they have left it like this. It seems like it's almost purposely confusing. Photosystem 1 is called that, photosystem 1, because it was the very first one discovered. It was discovered quite a bit of time before photosystem 2, and they've left it with that particular name. Photosystem 1 is called PS1 or also P700 because it accepts a slightly longer wavelength, 700 nanometers of light. Again, that would be still red, what we might call far red within our visible spectrum. So it's the second reaction center in the system, but it's called PS1 because it was discovered first. So let's kind of follow along with one electron or one particular set of electrons as they follow through this path. We're going to find out that there's two possible routes they can take. The main route that they usually take is called linear electron flow. That's the primary pathway through both of the photosystems and ending up creating ATP and NADPH using the sunlight energy. There is another way called cyclic. We're going to look at that in just a moment, but let's start with the linear, which is the main one. In linear electron flow, we have sunlight streaming down, making its journey over hundreds of thousands, millions of miles from the sun to the Earth. That's a journey of roughly 92 million miles, takes about 8 minutes. So that photon strikes the leaf, it hits a pigment that's located inside the chloroplast here. It excites the electrons within that pigment. That energy is passed from pigment molecule to pigment molecule until finally it gets to the inner portion of the photosystem here, and it excites the P680 molecule. The P680 molecule transfers that excited electron up to the primary electron acceptor, and we call it P680 plus because it has now given away that electron to the primary acceptor. So in the first step, we have light energy coming in, the photons coming in. It's exciting these pigment molecules. These pigment molecules are passing that energy throughout themselves until we get to the inner portion of the photosystem where that energetic electron is given to P680. P680 then carries that to the primary acceptor. The P680 molecule, the P680 plus, once it's given its electron away, it's a very strong oxidizing agent. As a matter of fact, it's one of the strongest oxidizing agents known in nature, and it really wants to replace that electron that it just gave away to the primary acceptor. So it will basically strip electrons from wherever it can get them. Now we have water that came in originally from the roots, and then that water was brought by the stem up to the plants. That water is going to be split by enzymes into hydrogen and oxygen. And those electrons are then going to be picked up to the P680 plus, the hydrogen atoms, which would include the proton and the electrons. So the P680 plus is going to see those hydrogen atoms and say, oh, that's what I need. It's going to grab it, and that's going to reduce it back to the P680, and it can be reused again and go through this cycle again. The other side of that, from the splitting of the water, is the oxygen. So we have H2O. We have two H's and one O. That one O gets released. It immediately combines with the nearest other single oxygen, and that becomes O2, which is atmospheric oxygen. That gets released. It leaves the leaf through the stomata into the air, and there you go. We get to live another day because the plants have created oxygen for us. Or I guess maybe not actually for us. It's a byproduct. It's something they got to get rid of in this particular case. So here's kind of the second step here. P680 has taken that excited electron, given it to the primary acceptor. Now P680 plus is like, hey, I really need some electrons, and there happen to be some electrons here as part of the hydrogen atoms that were split off from water. So it grabs that and it becomes P680 again, and the extra oxygen gets released and eventually leaves the leaf, leaves the plant. Those electrons that were captured in the PS2 photosystem are now going to fall down an electron transport chain from PS2 to PS1. And we're familiar with an electron transport chain. We've already seen that in respiration. It's what drove the proton pump, basically, so that it would increase the membrane potential. And then that allowed for ATP synthase to run as those hydrogen ions were passing through. Well, a very similar thing is going to happen here. We're going to use an electron transport chain to transport that primary, that excited electron from the primary electron acceptor of PS2 all the way down to PS1, which is the second photosystem. Just like we saw in respiration, this fall of energy, this release of energy as we go down the electron transport chain, that's going to drive a proton pump, which is going to increase the gradient across the thylakoid membrane, very similar to what we saw in respiration. And then that diffusion of those H plus ions, those protons coming back across to try to even out that gradient, that's going to drive that ATP synthase molecule to create ATP. So here we have our electron transport chain. We do have a couple of molecules in here that I may mention. We don't have to necessarily memorize them, but they are proteins that are part of the chain, including something called the cytochrome complex. These molecules are called plastoquinone and plastocyanin, and they are part of this entire chain of complicated proteins. But as this electron travels down this chain, energy is given up, and that energy goes to create ATP using ATP synthase, which is very similar to what happened in respiration. So now we're over here in PS1, which is the second of the photosystems. Just like in PS2, PS1 absorbs light energy, and it has pigment molecules that absorb that light energy. Just like in PS2, we have these pigment molecules get very excited, and they're carrying these excited electrons until eventually they carry it to the center portion there. They give it to P700. P700 then transfers that excited electron up to the electron acceptor, just like what happened in the other photosystem. And now P700 plus is looking for an electron, is missing an electron, and guess what? There's an electron that just came over from photosystem 2. So P700 plus now says, oh, hey, I'll just grab that one, and so it does. So here we are in photosystem 1, which is the second photosystem, remember. Light energy comes in, does the same thing, excites these little guys in here, these little molecules, and those little molecules are carrying the excited electrons eventually to P700. The P700 molecule carries that excited electron to the primary acceptor. Then P700 plus, because it gave up its electron, is looking for another electron. And guess what? There's one coming down the electron chain. So after it's passed this one, it goes and grabs that one that came down the chain from PS2. So we basically have an electron that's made a pretty long journey. It started out perhaps as the hydrogen part of water. Then that electron, along with its proton, it falls down an electron transport chain from the primary electron acceptor of PS1 to what's called a protein called ferredoxin. That ferredoxin is basically the end of the line. So we started in photosystem 2 with water. We went through the whole photosystem 2, then down an electron transport chain, and then we go through all of photosystem 1, and then finally we're going to end up at this protein called ferredoxin. Ferredoxin gives that electron to NAD+, which reduces it to NADPH, and that's pretty much the end of the process. So at the end of this process, we have NADPH, which are kind of like temporary batteries or buckets to hold that energy. And then, of course, we also created some ATP from the energy that we released in that fall down the electron transport chain. So the electrons of the NADPH and the ATP that we created now are going to be used in part 2 of photosynthesis, which is the Calvin cycle. This process also removes H+ ions from the stroma, and that kind of helps to reset that gradient that we were talking about earlier. So here's the entire process. Once these electrons make it up to the primary acceptor here, they're given to ferredoxin. Ferredoxin eventually gives those to a protein called enzyme called NADP reductase, which will create NADPH, and that's where we finally are going to end the first part of photosynthesis, which is with NADPH, which again is kind of like a little storage battery, a temporary battery holding that energy. So we've created ATP, we've created NADPH. Those two can be used in the next step. For me, in my mind, this analogy, this kind of physical, mechanical analogy here was a great help. It really helped me understand kind of the process of what's going on in the first step of photosynthesis, in the light harvesting step of photosynthesis. So take a look at kind of what's happening here. So we have a photon of light, and they're kind of using the mechanical model, but let's say that photon of light is imparting energy. That energy travels through all these different molecules, the different pigment molecules, until it makes it all the way up to the electron acceptor of photosystem 2. Then that electron is given to an electron transport chain. It travels down this path, giving up some energy, which is used to help create ATP. When that electron reaches photosystem 1, it's lower in energy. But remember, we have more photons coming in to add energy again, boost it back up to where it eventually gets to the electronic acceptor, and then finally that ends up being given to or using that energy to create NADPH, which is a bucket, a storage facility for that energy, which can now be used in part 2 of photosynthesis. So to me, this model I really liked. I thought it was kind of simple, but it was very clear, and it helped me understand what was going on. This model was quite a bit more clear than just looking at this. I had to flip back and forth a couple of times between these two to get a better understanding. Now, we did say there was another possibility for this electron flow, and this other possibility is not as common, but it's called cyclic electron flow. Cyclic electron flow only uses photosystem 1. Remember, that's the second one in line. And it does produce ATP, but it doesn't produce NADPH. Also, no oxygen is released, and that's because photosystem 2 is not being used. And this cyclic electron flow generates surplus ATP. So when you have a lot of Calvin cycle going on, which we'll talk about in just a second, you may need more ATP than can be created through the regular linear flow. So you may also have this cyclic electron flow going on in the background to kind of create more ATP. So here's a model of that, and basically we see that we're only using photosystem 1, and we have some of the same things going on, but from the primary acceptor we're going to ferredoxin, which is going back into the electron transport chain, and so we kind of have a cycle going on right here. This does create ATP, but because this photosystem 2 is not used, no oxygen is created, and we're not creating NADPH, we're only creating the ATP. So this is another method of electron flow, kind of a secondary method that can be used by plants. This cyclic electron flow is thought to be pretty ancient. There are some very ancient, simple organisms that do photosynthesis, such as these what are called purple sulfur bacteria. They have photosystem 1, but they don't even have photosystem 2. So they can only do this method. And it's thought that this is an ancient method of photosynthesis that evolved prior to linear electron flow. In modern day plants or further evolved creatures that do photosynthesis, it still has a purpose, because even though it's kind of considered like an evolutionary leftover, kind of like our appendix in human anatomy, it does serve kind of as a surge protector. When there are high amounts of sunlight, it kind of serves as a way to take that extra energy in bright sunlight conditions and use it and put it to use and help protect some of the rest of the system, because we're going to find out that some of the photosynthetic system is actually damaged by excessive amounts of energy, excessive amounts of light. So this is kind of a surge protector or an escape valve that allows that energy to be used in another way. We're going to end this portion by talking about what respiration and photosynthesis kind of have in common. And one of the things they have in common is electron transport chains. Another thing they have in common is the way they create ATP, and that's using this process called chemiosmosis. So let's look at chemiosmosis in chloroplasts and in the mitochondria. In the mitochondria, which is of course involving respiration, the electrons for chemiosmosis come from organic molecules. In chloroplasts, which is photosynthesis, the electrons come from the splitting of water molecules. So in the mitochondria, we're transferring chemical energy from food, say like carbohydrates or proteins or lipids, to ATP, whereas in the chloroplasts, we're transforming light energy into the chemical energy of ATP. So the source of the energy is different for these two particular organelles in these two different processes. The way that they are laid out, as far as the physical layout of the two organelles and the process that they go through for this chemiosmosis, there are some differences, but there's actually a lot in common as well. So it's interesting that these two processes occur in both of these different situations. Physically, some of the differences are that, say for example, in the mitochondria, the protons, the H plus protons, are pumped into the intermembrane space, and then as they flow back through into the mitochondrial matrix, that's what drives ATP synthesis. In chloroplasts, those protons are pumped into the thylakoid space, and then as they diffuse into the stroma, they drive ATP synthesis from the ATP synthase, which is located in the thylakoid membrane. So here's kind of a comparison of them. So you see the darker gray represents a higher amount of the H plus ions, and the lighter gray or white represents a lower H plus ion concentration. So the same thing is occurring in both of these mitochondria and the chloroplasts. We have an electron transport chain that helps pump H plus ions into a particular area, the intermembrane space of the mitochondria and the thylakoid space of the chloroplast. And then as they automatically diffuse down, trying to take care of that gradient difference, they go through the ATP synthase, and that powers the creation of ATP. So it happens in different places in these two structures, but the actual process is pretty much the same. In the chloroplasts, so for the light reactions, the ATP and the NADPH that we've created, they're produced on the side of the thylakoid membrane that faces the stroma, because that's where the Calvin cycle is going to take place, so that's where those molecules are needed. So they are created and produced and basically spit out on the stroma side of the thylakoid membrane, and that's where they're going to be put to use. So if we had to summarize the first portion of photosynthesis, it's all about gathering light and using that light to create energy-holding molecules. So light reactions generate ATP, and they increase the potential energy of the electrons by taking them from water, where those electrons are at a lower state, and moving them up to NADPH, where they're at a higher state. It requires energy to get that done, and oxygen is given off as a byproduct. So then, what are we going to do with that energy? So now we have energy in the form of ATP molecules, we have energy in the form of NADPH. How are we going to put it to use? Because we haven't created any food yet, and I'm sure you thought that photosynthesis was all about creating food for the plant. So all we've done is capture light energy, but we haven't really made anything with it. We haven't made any food. So the next step would be to take that captured light energy and to actually create some glucose to create some food to feed the plants, which would eventually feed us. So we're going to stop right there, and then we'll go on to part 2 of photosynthesis. Great little overview here of the first part of photosynthesis. We are headed to the Calvin cycle. So notice this is in the thylakoid space, and we have photosystem 2, photosystem 1. All of this is tied together. We're creating NADPH. We also are pumping protons to change the gradient between the inner and outer portion, and that's going to allow for the production of ATP. Those two molecules, NADPH and ATP, are going to head off to the Calvin cycle, part 2.